In this study, we investigated the distribution of cells expressing vomeronasal receptors in the olfactory organ of turtles. Many tetrapods have two olfactory organs, the olfactory epithelium and the vomeronasal organ. In mammals, the OE lines the dorsal-caudal portion of nasal cavity, while the VNO is situated at the base of nasal septum. The olfactory organs contain olfactory receptor neurons, which extend an axon basally and a dendrite apically. Generally, the olfactory receptor neurons in the OE bear cilia at the tip of their dendrites, while those in the VNO bear microvilli at the tip of their dendrites. Olfactory chemoreception is mediated by the olfactory receptors categorized into three families: odorant receptors, type 1 and type 2 vomeronasal receptors. They are coupled to G-OLF-GI2 and GO respectively. In mice, olfactory receptor neurons in the OE express ORs, whereas those in the VNO express vomeronasal receptors. In the VNO, olfactory receptor neurons in the apical layer express V1Rs, while those in the basal layer express V2Rs. In fish, a discrete VNO does not exist, and the OE contains both ciliated and microvillous olfactory receptor neurons. As in the case of mammals, ciliated neurons express ORs and GOLF, whereas microvillous neurons express V2Rs and GO. Thus, it is believed that the fine structure of olfactory receptor neurons and the gene expression of olfactory receptors are closely related to each other, and this relationship is conserved among vertebrates. The olfactory organ of turtle is comprised of two types of sensory epithelia: the upper chamber epithelium, which lines the dorsal portion of the nasal cavity, and the lower chamber epithelium, which lines the ventral portion of the nasal cavity. In general, the olfactory receptor neurons in the UCE bear cilia and microvilli, and thus the UCE is regarded as the OE. Meanwhile, the olfactory receptor neurons in the LCE bear only microvilli, and the LCE is regarded as the VNO. Based on the fine structure of olfactory receptor neurons, it is speculated that the olfactory receptor neurons of turtles in the UCE express both ORs and VRs, while those in the LCE express only VRs. In order to elucidate the olfactory receptors expressed in the olfactory organ of turtles, we analyzed the expression of VRs in the present study. Red-eared sliders and soft-shelled turtles were used. In situ hybridization was conducted using dig-labeled CNA probes prepared by PCR cloning. The density of cells expressing VRs and TRPC2, as well as their spatial distribution, was analyzed. In turtles, V2Rs are encoded by two genes, including V2R1 and V2R26. Cells expressing V2R1 were found only in the LCE, while those expressing V2R26 were found both in the UCE and LCE. The density of cells expressing V2R26 was higher in the LCE than in the UCE. In the olfactory organ of soft-shelled turtle, as in the case of red-eared slider, the density of cells expressing V2R26 was higher in the LCE than in the UCE. The V1R is encoded by two genes, V1R3 and V1R14. By the RT-PCR, the expression of V1R3 was demonstrated in the olfactory organ, but that of V1R14 was not. V1R3 gene was found to be expressed by cells both in the UCE and LCE. Also, the gene encoding TRPC2, an ion channel mediating the signal transduction VRs, was shown to be expressed by sparsely distributed cells both in the UCE and LCE. Spatial distribution of cells expressing TRPC2 closely resembled that of the cells expressing VRs. In contrast, the gene encoding CNZ2, an ion channel mediating the signal transduction of ORs, showed extensive expression both in the UCE and LCE. By analyzing the expression of VRs in the olfactory organ of turtles, we found only a small portion of olfactory receptor neurons express VRs. In contrast, ORs were extensively expressed by the majority of olfactory receptor neurons. This indicates that the correlation between the fine structure of olfactory receptor neurons and the gene expression of olfactory receptor is not conserved among turtles and other vertebrates. This study received Outstanding Paper Award in 2020 JVMS by the Japanese Society of Veterinary Science.